Hi, everyone. So in this talk, everything at the core of it is about diffusion MRI. And so diffusion MRI, we use it as an excellent probe of tissue microstructure. The way we do this is that basically in a diffusion MRI experiment, the signal is sensitized to this random displacement of water molecules, or we can also do it for metabolites. And it so happens basically that in the diffusion time that we impart, the displacement of these molecules is on the order of a few tens of microns, which actually matches the typical length scale of the cellular layout we're trying to characterize indirectly. And so the path of these water molecules will probe the microstructure by bouncing off membranes or experiencing hindrance and restriction. And so we can glean a lot of information on micron scale features of what happens at the cellular level, although the voxel size is rather on the order of the millimeter. And so in the group, we have two lines of so-called microstructure research. The first one is to develop models for tissue that then give us access to specific features of this microstructure by fitting the model to the signal. And so we call this mapping the substrate. And so we develop methods for quantifying tissue microstructure in brain, white, and gray matter, but recently also in other organs outside the brain. And the other part of our research is about exploiting dynamic fluctuations of microstructure with brain activity to actually map the information or the traffic that travels through the brain. And so to this end, we developed diffusion functional MRI as a complementary fMRI contrast to the more typical bold that relies on hemodynamic coupling. And so in this research, we have to walk through a certain number of steps and pipelines. So we start off with simulations, be they analytical or numerical. Then we spend some effort in the parameter estimation or so-called fitting that can be done with traditional algorithms or machine learning. And then, of course, when you implement something experimentally for the first time, it's very convenient to do it on preclinical scanners because you have a lot of flexibility there in terms of the protocol you can set up or the scan time. You can do some validation against histology. And of course, there are animal models of disease that are very interesting. Then we move on to clinical research data acquisition. So this is typically our reality check. This is when you try to do something in human for the first time. Things can become complicated because the hardware is not as proficient. You have limited scan time, et cetera. So very different hurdles to overcome. But once a method is robust, of course, we're very interested in translating it to patient cohorts and then trying to understand how the microstructure changes with disease, trying to look at interactions with other markers you may be able to measure. And we're also invested in developing a normative modeling approach to brain microstructure in the human. And so in order to go through all these steps, I have to say we very largely benefit from the local MRI landscape that is made possible by the CIBM. So in radiology, we have access to human scanners, 3 Tesla, and low field. But when we need to, we also have, of course, the animal scanners that are available to us at EPFL. And when we need, for example, higher SNR or spatial resolution, we can even go to the 7 Tesla in Geneva or their Prisma system that has features that we don't have in radiology. And more recently, because in diffusion, a lot of it is about magnetic field gradients, we're also looking forward to using the SIMA-X with the stronger gradients in CHUV radiology. So all of this really opens up a lot of possibilities. And when those gradients in the local area are not enough, we sometimes even travel to Cardiff or Boston for the high-end connectome human systems. And so I will try to really praise this environment here and everything we've been able to do using this infrastructure. So let's get started on white matter microstructure. So over the years, the community really converged to establishing this white matter biophysical, a so-called standard model that is made up of 2 compartments. So roughly, we consider the intraaxonal space. So these axons are modeled as sticks with some diffusivity along the axon. And they occupy a certain volume fraction here in the voxel. And the rest is extraaxonal water that can be characterized by 2 diffusivities, one along the fascicle, the other perpendicular. And these fascicles then have some orientation distribution function to them to model how dispersed or aligned they are. The reason these parameters are important as compared to, for example, to something like diffusion tensor imaging, which you may be more familiar with, is that the hope is that they can bring more biological specificity in our non-invasive characterization than empirical markers. So typically, we can develop methods that give us more specificity to axonal density, pruning, axonal integrity or injury, myelination, demyelination, and also inflammation. And so this model has undergone a large number of validation steps that I will not go in detail today. The point is that it's actually clinically feasible. And so I want to actually present some applications already of this model in disease. One thing I want to mention is that in order to estimate these parameters, you have to move away from the typical single shell DTI scan, so a single B value with a bunch of directions, and you really need to go at multiple B values, and the minimum there is 2 shells, basically. But there are many applications of this white matter standard model. This is an example here in stroke. This is a work from NYU, where you see basically in the stroke area, the parameters that most capture the change with stroke are this intraoxonal water fraction that increases dramatically, and you also have a dramatic drop in the intraoxonal diffusivity. And this is very specific to what happens in stroke, which is cellular swelling and massive axonal beating that would really make this diffusivity along the axon drop. And so then you may wonder, okay, is it always the same parameters that change with pathology? Well, actually, no. Depending on the pathology, we do have specificity. And so in the group, we've worked on this Lausanne psychosis cohort that was part of the NCCR synapse. So this is collaboration with the Center for Psychiatric Neuroscience. And so comparing the early psychosis and schizophrenia patients to age-matched controls, we find that here the differences in the pathology are driven by a drop in intraoxonal water fraction, but actually, most importantly, an increase, this time in the extraoxonal diffusivity. So different compartment and different trends for the parameters. And this also makes sense with what is known of psychosis, that is actually adolescent early on changes in glial density, extraoxonal changes, and myelin defects. So we can start to be quite specific about the things that we can characterize non-invasively in these patients. We wanted to take this one step further and also see how this white matter microstructure change correlates with other markers of the disease. So here looking at metabolite concentrations in the frontal cortex. So this is a collaboration with Li Jingqin, who did the MRS analysis. And here actually we found that only in the patients we had microstructure parameters, so this intraoxonal water fraction and the extraoxonal parallel diffusivity, the same ones that drove the group differences here, that were also correlating with metabolite concentrations. And they did that in the white matter tracts that were also connected to this frontal part of the brain where the MRS measurement was taken. And even more importantly than that, the only metabolite that correlated with white matter microstructure changes was myoinositol. And myoinositol is a glial marker, so this really establishes a very clear link between glial density and function and the white matter extracellular diffusivities and how the white matter environment is affected as a result. Okay, so going further with this standard model, this time at 7 Tesla, we're trying to connect these microstructure features to something like conduction velocity in the human brain. And here we went to 7 Tesla to try to get higher spatial resolution and benefit from the higher SNR. And so we have, for now in healthy controls, these microstructure maps, to which we also added the estimation of the axon radius using a diffusion relaxation model developed by Barakovic and colleagues in Jean-Philippe Tirant's group. And so combining these microstructure features, basically we measure them along different white matter bundles. And in parallel to that, we have a measurement of conduction velocity in some of these bundles by combining invasive EEG measurements of delays with fiber length. This F-track data set is made available by Aix-Marseille. And so now we try to see how the microstructure can predict conduction velocity. And we actually found that the parameters that best act as predictors for conduction velocity are, again, axonal water fraction and axon radius, which also makes sense with previous simulation models that were trying to explain how the axon morphology would impact conduction velocity. And the nice thing about this is that now we can build a predictive model to paint in basically the missing information here. So you cannot put EEG probes everywhere, okay? So there are some white matter bundles for which we don't have the direct information, but we can now fill it in indirectly using the microstructure-derived model. And the goal ultimately is then to also apply this to epilepsy patients. And this project is a collaboration with Serge Villemot and Dimitri Vandeville, as well as Patrick Hagman at CHUV. Okay, so let's move on to gray matter. So now, well, different tissue type, different model. We have to start from scratch. And so what is a relevant model for gray matter? What differentiates it from white matter is certainly the amount of cell bodies. So maybe we have to model this now as a third compartment. Or the fact that there is a lot less myelin, so maybe the structures now are not impermeable as they are in white matter, and maybe we have to account for this exchange across the cell membrane. And unfortunately for now, numerically, it's basically impossible to model everything at once. The fit or the model is unstable. But we realize that if we have to choose, the most prominent signature that we get from a measurement in the cortex is actually that of exchange. So it makes more sense if you need to choose to model the exchange and leave out the cell bodies for now. So this is what we've decided to do. And we proposed this NEXI two-compartment model for gray matter for neurite exchange imaging that accounts basically for similar features as the white matter. So you have the cell process density, intraneurite diffusivity, extracellular diffusivity for inflammation, and now this exchange time that can be a good proxy for cell membrane permeability that is probably modulated by myelination or membrane dysfunction. Little bit of a warning here, in order to estimate this, not only do you have to go multi-shell, so different B values, you also have to go multi-diffusion time, so changing the duration here between the separation of the gradient pulses. And so this makes for quite advanced and lengthy protocols, but this is something we're also working on reducing. And so, of course, you try out the first thing, a model for the first time. It's easier to start on this 14 Tesla at EPFL in animals. And so we did this NEXI study in the rat brain a few years ago where we had a lot of flexibility in choosing the B values and the diffusion times, and we produced these quantitative maps of the gray matter in the rat in vivo. And then we also did some comparison with histological staining. So here, for example, for neurite density, in the cortex, we have this low, high, low pattern along the cortical layers, which was nicely matched by the staining for neurofilaments. But in the hippocampus, if you look at the different subfields, the contrast actually matched better with the staining for astrocytes. So what we learned from this is that when we say neurite density, what we really mean is cell process density, because these astrocytes, they have extensions that kind of agree anatomically with the neurites from the neuronal cells, and because water is everywhere, we don't have cell type specificity, so we're basically sensitive to both. So this is also something we have to account for in the interpretation. Of course, any model is only worthy if you can actually ultimately apply it to human, and so this was basically the entire PhD thesis of Quentin Hull, trying to move the NEXI model from a preclinical grade scanner to human scanners. And so we did this in steps. Initially, we went to Cardiff on their Connectome 1 scanner to still have a lot of gradient strength and some flexibility here in choosing the protocol. Already at this step, we learned that in our model directly, we have to account for the Reichen noise floor if we have lower SNR than we did on the animal scanner, so this has to be built in. And then we finally took this also to our Prisma scanner at CHUV, compromising, of course, always on the B values and the shortest diffusion time given the available gradient strength. And here what we learned is that we have to also account for the long gradient pulse duration. So when you don't have amplitude, you have to make your gradient pulses fatter. And so this has to also be accounted for in the model. But so gradually, we really managed to obtain these quantitative maps of a human cortex microstructure in vivo. And so comparing this distribution, for example, to that of myelin density obtained in the literature, we can see that we also find a higher density in the central sulcus here, as well as a longer exchange time, which agrees with higher myelination. However, it's not a one-to-one correspondence. Here in the temporal lobe, you see we have longer exchange time that is not explained by myelin. So it could also be I don't know, glial to neuron density balance, or maybe spine density. So this is something that we're also currently investigating. There are still open questions. But this framework is available open source if you want to try it on your own data. And so in humans, well, you cannot really have histology on in vivo subjects. So what we tried to do at least is a cross-validation with other MR contrasts. And so we looked at two myelin contrasts. One is this myelin water fraction using a multi-echo T2 acquisition. And so we were very happy to find that among the NEXI parameters, the one that correlated best with this myelin water fraction was the exchange time. So there is a good agreement and sensitivity there to myelination in the cortex. And it was the same story when we did inhomogeneous magnetization transfer, which is also specific to myelination. Again, the exchange time was the one that correlated the best with this IHMT ratio. And so moving on from this, of course, we want to do more with less, reduce the scan time. Initially, we were at 30 to 40 minutes, which is really prohibitive. We were able to reduce it in half by doing some kind of informed reduction of number of measurements that we were taking. And also, we want to further model both exchange and soma, and this is ongoing work from many groups across the world, but for now, it's a difficult problem. There are many applications possible. In the mouse and rat world, we're already working on a GLUT1 deficiency syndrome in the mouse and on a rat model of creatine deficiency as collaborations with the Center for Psychiatric Neuroscience, and here with Olivier Bresson and Cristina Godalbo. And in humans, I think the most obvious applications would be multiple sclerosis and traumatic brain injury, and this is also something we look forward to pushing in the near future. Now, in terms of this validation of the different models, we typically try to use histology, electron microscopy, other MR contrasts. They are nonetheless never but silver standards, because if you've ever done histology, you know it's not such a ground truth. It's a good comparison, but each modality has its limits. And when you do validation with analytical forward models, I mean, all you do is noise propagation. It's a bit of a circular argument. You generate your signal with a model, then you fit it with the same model. Of course, it works. And so we wanted to generate ground truth where we're able to deviate the reality from what the model actually accounts for and see how robust it is. And so this brought us to developing basically synthetic cells and synthetic substrates where we can grow them with tunable cell complexity so they can have undulation, beating, spines, etc., and then packing them in these synthetic voxels where we can do Monte Carlo simulations of random walk. And so this is now used to test the accuracy and precision of the white matter and gray matter models, and also of metabolite diffusion models, here also in collaboration with Cristina Godalbo. And for this, we used this MCDC simulator that was also presented by Pina as software made available through CIBM, and so we collaborate strongly here with Johnny from Jean-Philippe Thirand's group. And so if you're interested in these techniques, there are 2 posters in the hall touching on these substrates and the simulations. Okay, just a quick slide about outside the brain. It's important to me to mention this because sometimes it's also the environment that drives the projects that we take on. And so in radiology, we have 2 very research-savvy and driven abdominal radiologists who are Clarisse Tromin and Anaïk Vietti-Violi. And so it's really their enthusiasm for research that also brought me on board of these projects of diffusion outside the brain. And so the first one we are working on is this biparametric prostate MRI protocol at low field on the FreeMax. And so the goal here, of course, as also Margaret pointed out, prostate cancer is a huge societal concern. And so being available to do prostate screening on a low field scanner, which is cheaper and maybe more available down the road, is very important. And so we wanted to propose a PIRADS compliant protocol for this screening at low field within still short exam time. Now, if any of you have seen low field images or presentations, you will know for now it's almost inseparable from using deep learning reconstructions to mitigate the loss of SNR. And here there is no exception, right? So this is our reference T2 weighted image of the prostate at 3T. If you don't know what you're looking at, the prostate is here. And so at low field, the raw image is of course much grainier, but using the deep learning reconstruction from Siemens, it's actually, the quality becomes comparable to the 3 Tesla, again, in a similar scan time and similar resolution. Now for diffusion, which is the other contrast that comes into this PIRADS evaluation. So this is an ADC map of the prostate, apparent diffusion coefficient of the prostate at 3 Tesla. And playing a bit on the parameters and trying to optimize it on the low field, we also managed to get a decent ADC map, again, in comparable scan time. So we're very happy with this, and so now we're deploying it in patients to try to see if we can also detect lesions as reliably as at 3 Tesla. And then for the 3 Tesla abdominal scans that are more your run-of-the-mill, what we're working on is really extending the diffusion MRI protocol. So typically people clinically would just look at this ADC using moderately high B values. We're interested to see if we push the B values and also estimate non-Gaussian effects such as kurtosis, do we glean more information in characterizing disease? And so we're working on, for example, on liver fibrosis, using diffusion as some kind of virtual elastography measurement, and then stratifying patients as a function of their fibrosis stage. And also we're looking at extended diffusion MRI acquisitions for characterizing esophageal cancer and seeing the prediction to treatment response. And there's also a poster about this in the hall if you're interested to check it out. So in the few minutes I have left, I just want to walk you also through our endeavor of a diffusion functional MRI to try to map traffic in the brain. And so to start off, before I even go into sort of any MRI measurement, I just want to mention that with optical techniques, people have reported microstructure fluctuations with neural activity. So here you can see that the neuron cell body here, the soma is displaced temporarily, so are the dendrites. And people have also shown this in the white matter, actually, that there is a transient swelling in the myelinated segment of the axon as the train of action potentials is going through, that then goes back to baseline. So this effect is actually real. And so because diffusion MRI is so exquisitely sensitive to this microstructure, the question is, can we use it as a functional contrast, and particularly in the white matter? So why the white matter? Well, because we actually don't have, for now, a good neuroimaging technique that gives us traffic or activity in the white matter. So EEG and MEG are sensitive only roughly to the cortex. They are limited in depth. And the bold functional MRI contrast relies on this neurovascular coupling, and therefore the sensitivity is limited by vasculature. And so you have less vasculature in white matter, a very weak and very altered bold response. And so there was really somehow a gap there that could be perhaps filled by diffusion functional MRI. So how does diffusion functional MRI work? I mean, you still need a time course of measurements, right, to detect fluctuations. So what you need to do is to do repeated acquisitions of diffusion-weighted images. And so you have to acquire 2 different B values in alternating fashion in order to be able to calculate an apparent diffusion coefficient map at each time point. And so there are different steps that you have to optimize. This ADC time course allows you to minimize T2 weighting that is intrinsically built in these diffusion-weighted images because they are spin echo-based. And if you're T2 weighted, you will capture bold contrast, so you do not want that. So the ADC calculation is really paramount to keeping bold contrast out of your time course. We want to work with B values that are high enough, again, to suppress as much as possible direct blood contributions. And finally, we even have to optimize the waveform to try to mitigate cross terms between our diffusion gradients and the background bold susceptibility gradients. But if you do this, basically, so this is what we call careful sequence design. So with this careful sequence design, you can measure ADC time courses that are minimally affected by bold contributions and give you this very nice ADC drop upon the task that is different then from the bold response. And now if we want to look in white matter, we have another problem that is orientation dependent. So if you sensitize to diffusion in one direction of space at a time, people already reported quite a few years ago that if you're perpendicular to your white matter fibers here in the mouse optic nerve, you can very robustly detect this drop in ADC. But if you are parallel to it, you almost see nothing. And in the human brain, I mean, white matter tracks, they take all sorts of orientations. So if you choose one, basically you really have anisotropic sensitivity to activation across the brain. So this is not what we wanted. So how can we achieve isotropic sensitivity? So for this, we turn to this spherical B-tensor diffusion encoding. So this would be your typical linear encoding with your two diffusion gradient pulses. And if instead of those, you play some pre-calculated funky waveform, basically you manage to sensitize to diffusion in all directions of space in one shot, okay? So this is not an average of different directions. In one shot, you sensitize to diffusion in all directions of space. And so this is the choice that we made. And so using this kind of approach in visual task fMRI study, we now report activation clusters along the optic radiations with ADC fMRI that are not matched by clusters that you can detect with BOLD, that really, of course, covers, as expected, the visual cortex and the relay in the thalamus here. So there is really something complementary about these two techniques that I think can be further exploited. And so we're working on this further in terms of characterizing the contrast. We really wanted to make sure we are independent from vascular signals. And so we designed a breath hold study. So breath hold will cause hypercapnia and vasodilation. So you'll have a vascular response without underlying neural activity. And so if you correlate the end-tidal CO2 measurements to your various functional MRI time courses, of course you have dramatic correlation with BOLD fMRI because it's a vascular response. And you have almost no correlation in ADC fMRI, if not maybe in the CSF here. So this was very reassuring. Now we're also working on looking at this, the results of such visual stimulation in the rat on the preclinical systems at EPFL. There's also a poster about this if you're interested to check it out. And then in terms of applications, well, one that we think is maybe interesting is using it for resting state functional connectivity. So here what we can do is that we can extend this connectivity matrix not to just accommodate gray matter nodes as it usually would, but also white matter nodes, and try to look at white to gray and white to white functional connectivity. And so doing that with BOLD fMRI versus ADC fMRI, we basically found that we can retrieve the typical gray to gray connectivity that you would get with BOLD, but in addition, we have stronger white to gray and white to white connectivity that we can find with ADC fMRI. So I think from a neuroscience perspective, this is possibly interesting and something we're further working on at the moment. Okay, that's all I wanted to share with you today. I want to thank my group and all the collaborators across the different institutions, and thank you very much for listening.